Okay, so up to now, what we see is that we take in mind that microbiome growth is so bad, is so negative. Not always. Some cases, microbiome growth is what we want, desirable. In fermentation, like I mentioned a while ago, in making nemcure, fermented sausages, we need the lactic acid bacteria to grow, okay, to make the product. We want them to grow. So in this case, we don't inhibit them, but we know how to stimulate them. We inhibit the other undesigned microorganism, the one that we don't want them to grow, but we need to stimulate lactic acid bacteria. In curing meat, là ướp thịt, we also need lactic acid bacteria to convert nitrate into nitrite. Nitrate is like NO3, we convert into NO2. We convert nitrate to nitrite to do function in curing meat to make the red color to prevent other microorganism to grow. I think like we will discuss more in detail in the next semester. Right, and now we go to the methods that we can apply to control or to destroy microorganisms. So the first technique or the first approach, cách tiếp cận, is by using preservatives. Preservative là chất bảo quản. Okay. The additives, the preservatives are the allowed chemicals, should be allowed chemicals which inhibit the growth of microorganisms. For example, the allowed preservatives are nitrate, nitrite, sulfur dioxide, benzoate, and things like that, depending on the products. And when we use preservative, what we need to do is we have to use the approved preservative, the allowed preservative. And the second important point we need to pay attention, we have to follow the allowed dose. Each preservative, the regulations là quy định về thực phẩm, food regulations only allow certain dose to be safe, because preservative inhibit or kill microorganisms, then they are also harmful to our cells, to our body, if we do not follow the dose. So we have to follow the dose. When the dose or the concentration, I mean the concentration is below a certain level, it would be safe. But when you abuse, violate, then it's not safe anymore. So you have to follow the dose strictly. And not only that, you need to make sure that you mix into the product, dissolve into the product homogeneously, evenly. You need to mix as well. Like for example, if you have a batch of the product, if you only put preservative here and you don't mix as well, here it's become overdose. Overdose. It's become not safe in terms of the dose of the preservative, and here it's become underdose and the spoilage still occur, the pathogens still grow. Not good. To be sure that we apply the ways that's distributed chemical evenly. Now the second approach is to use heat treatment. Heat treatment includes pasteurization. Pasteurization then we carry out in the range of around 63 to 100 degrees C. Then we kill vegetative cells. Vegetative cell means living cell. Okay, the living cell are pathogen, tế bào sinh dưỡng. We focus first on the pathogens. The main objective, the main target first is the pathogen, but not spots and the vegetative cell of the pathogen to make the product safe first. Okay, safe for human consumption mean will not have food poisoning. This is the first objective, là cái mục tiêu đầu tiên. And the second objective is also to destroy a main part, important part of spoilage microorganisms. Spoilage mean they do not cause food poisoning, but they cause spoilage of the food. Will be expired, no more uses, and the color will be not okay, smell will not be okay, texture will not be okay anymore. So we need also to destroy spoilage microorganism to extend the shelf life. The temperature is not high enough, so this process cannot destroy spore. Sterilization, then we apply the temperature above 100 degrees C. We will study detail somewhere in what range of temperature, but now we know it's when it's above 100, so then we can also destroy the spores, about those which are very resistant to heat, so you need high temperature to kill. For heat treatment, after that, what we need to pay attention is the cooling. Especially in meat processing, we need to carry it out cooling properly. Otherwise, you also have issue. For example, you have a large piece of meat like this, and you cook. You cook when you do pasteurization, means that you do not destroy all microorganisms. You see part of, not all. Even vegetative cell, you do not destroy all. Spore, you do not destroy any. So after cooking pasteurization, if you don't cool it fast enough, within here it still be warm for a long time, and then the survived microorganism can grow fast after that. This is not good. Okay, the third approach we can apply for control or for destruction of microbe is by using packaging system. There are different techniques of packaging. Vacuum packaging. Vacuum packaging means the way before we pack, we remove all the air. When we remove all the air, then of course you remove oxygen. You remove oxygen means that you reduce the growth of aerobic microorganisms like mold and some bacteria. Not only that, you reduce the oxidation of food. So, for example, if the product has oil and sensitive to be oxidized, nó bị oxy hóa, nó bị ôi dầu đó, fat rancidity. So, if we remove the oxygen like this, you can reduce the oxidation and the cell life in terms of chemical reaction. This is microbial changes, thay đổi về mặt sinh học, sinh vi sinh vật, thay đổi về mặt hóa học này. Những cái thay đổi từng cái nhóm như thế này nó có thể ảnh hưởng đến hạn sử dụng. Thì vacuum nó sẽ làm được hai cái việc này. Okay, so vacuum packaging can reduce the growth of aerobic microorganism, can also reduce somehow oxidation, which is a chemical reaction, and then can extend the shelf life. Modified atmosphere packaging, MAP. I mentioned a little bit a while ago. So instead of using air, normal air, we change the air composition. We modify the atmosphere. So we also can have similar function. The difference between vacuum packaging and MAP is that for vacuum packaging, you remove all the air, so the product is quite collapsed. The package is really like shrunk, shrunk like co lại đó. It's contracted into the food like that. For this one, you don't do vacuum, so the pack still look okay. And we can apply active packaging, like we can use the oxygen scavenger, things like that. We can put some material inside the package, and this material will absorb all the oxygen, or sometimes like humectant mean that we absorb the uh, sorry moisture absorber that absorb the vapor also. So it depends what function, what objective we need to apply. Here we can use oxygen scavenger, mean the material that catch all the oxygen inside. Or canning sterilization is also a way of packaging.